COVID-19 has highlighted the need to manage healthcare resources as effectively as possible. Intensive care consumes enormous amounts of resources and is one of the most expensive aspects of healthcare globally. Evidence-based treatment in intensive care can significantly reduce healthcare costs.
We deal with the sickest and most vulnerable patients in hospitals. Our highly skilled medical and nursing teams look after people who require vital organ life support following severe injuries, major surgery, and those with acute organ failure due to severe illnesses and infections. The COVID-19 pandemic has brought these kinds of patients into the public spotlight and highlighted the immense efforts required to keep these patients alive. We base our treatment choices on the best clinical research evidence available. When there is uncertainty about the best evidence, there is an imperative for us as clinician researchers to generate new research of the highest validity.
So just about every patient admitted to an intensive care unit will be given intravenous fluids at some point. But which fluid produces the best outcomes has actually long been debated.
Intravenous fluids containing albumin were traditionally used very commonly in Australian intensive care units. But a 1997 study suggested that such fluids could increase the risk of intensive care patients dying. We felt we had an ethical and scientific duty to find out if this was true, but doing so needed us to conduct a trial of a scale never before attempted in intensive care units. So from 2001 to 2003, I led a trial of 7,000 patients across Australian and New Zealand intensive care units to compare the effects of albumin with a cheaper fluid, saline. We found that albumin did not improve outcomes for critically ill patients compared with saline. In fact, its use increased the risk of patients with traumatic brain injury dying. Our findings gave and continue to give clinicians robust and compelling evidence. The routine use of albumin can no longer be justified.
Across the world, where albumin solutions were not readily available or were too expensive, alternatives such as hydroxyethyl starch solutions or HES solutions were widely used. This practice developed despite a high amount of uncertainty about the safety and efficacy of HES solutions in high-risk patients, particularly those with sepsis. To address this lack of evidence, we conducted another large-scale randomized control trial among 7,000 intensive care patients, similar to the previously mentioned albumin study. We found that HES solutions did not improve outcomes compared to saline, and in fact increased the risk of patients requiring acute dialysis for kidney failure, particularly those patients with severe sepsis. The study findings led to major changes in clinical treatment guidelines worldwide. The global impact of this research can be realized by the thousands of lives and millions of dollars saved by preventing the exposure of high-risk patients to HES solutions.
So our research has changed the way the medical world thinks about intravenous fluids and showed that the choice of fluids should be treated with the same care and attention as the prescription of any other drug. We have just completed a trial of over 5,000 patients to determine whether saline or the increasingly popular buffered or balanced salt solutions produce better outcomes for intensive care patients. The data from our fluid trials will continue to influence clinical guidelines and practice worldwide and will improve the health of millions of acute and critically ill people.
By tackling what was previously considered impossible, our researchers launched a new era in intensive care medicine. Their findings have influenced intensive care worldwide, prevented commonly used but harmful treatment practices, and demonstrated that cheaper treatments are often safer than more expensive ones.